Okay, so welcome back to this next video in which we are discussing opioid receptors. Okay, so we've talked about the four different types of opioid receptors now. The mu opioid receptor, the kappa opioid receptor, the delta opioid receptor, and also the opioid receptor like 1, the ORL1. Okay, and basically we've discussed that all four of these are rhodopsin family G protein coupled receptors, which means that when their ligand binds to them, it binds to residues which are within the transmembrane domains. Okay, and causes a conformational change in the G protein coupled receptor that then allows the G protein coupled receptor to bind with the heterotrimeric G protein. Okay, so we want to now discuss the heterotrimeric G protein, or G proteins, which these four opioid receptors can bind to. Okay, so basically we need to discuss how many different heterotrimeric G proteins there actually are. And to do this, we need to look at how many different alpha subunits the human body has. We need to look at how many different beta subunits the human genome has, and how many different gamma subunits the human genome has. Okay, so basically you have 16 genes which code for different alpha subunits. Okay, so we'll start off with the alpha subunits. So there are 16 genes coding for different alpha subunits, okay? And when I say we have 16 genes, what I mean is discounting the homologous chromosomes. So I'm not talking about the fact that you have 2 copies of each gene. Okay, I'm talking about if you just look at one copy of each of the homologous chromosomes, you will find 16 genes for alpha subunits on those copies, on those chromosomes basically. Okay, if you then take into account the homologous chromosomes, you'll then have 32 genes. Okay, so they'll double up. But we're just talking about one copy of each homologous chromosome. Okay, now, how many alpha subunits can you actually produce from these 16 genes? Well, actually, you can produce 21 different subunits. Now, this seems like a bit of an anomaly, but the reason for this is that some genes have multiple known splice variants. Okay, so let me explain what I mean by that. So basically, let's just go over the central dogma of biology. So let's let these two lines here represent the double-stranded DNA, basically. Okay, now if we're looking at the gene for an alpha subunit, then you will have a coding strand, okay, and you will also have a non-coding strand. So I'll show the coding strand in turquoise, and I'll show the non-coding strand in red. Okay, so in turquoise, this is the coding strand. Okay, and in red, this is the non-coding strand. Now, what does this actually mean? Well, basically, when you actually produce mRNA from the DNA, you actually only need one of the strands of the DNA. You don't need both of the strands of the DNA, because you're producing an mRNA molecule which is complementary to one of the strands of the DNA, and the strand that you actually use is the coding strand. Okay, so basically you will produce a piece of mRNA which has a complementary sequence of organic bases to the sequence of organic bases of the coding strand. Okay, so this other line that I've drawn parallel to the coding strand, this represents the mRNA, and I've now highlighted it in vivid purple here. Okay, and it is RNA polymerase which will produce this mRNA from the coding strand. Okay, so here then now is this piece of mRNA in vivid purple here. Okay, now, basically, this is what would be called pre-mRNA. Okay? This is not actually ready to be translated yet. Okay, so if we put this through a ribosome and read it and produced a sequence of amino acids in a polypeptide which was coded for by this, it would make a nonsense protein, basically. What we first have to do is cut this up and then cut certain bits out, because basically you only actually want to use certain portions of this pre-mRNA to actually be translated into a protein. Okay, so let's mark these out. Okay, so the portions that you are actually going to use in the translation process are called the exons. So let's call these the exons that I've now highlighted up here. Okay, and the portions of mRNA in between the exons are then called the introns, and you don't want to use these portions. And this picture is just to illustrate the point. It's not saying that any of the alpha genes specifically have 3 exons and then 2 introns. It's just to illustrate the point. Okay, so the point is that when you produce the primary transcript, or the pre-mRNA, which is just directly complementary to the coding strand of the DNA of the gene, basically some of the portions of this mRNA you don't actually want to use when you translate the mRNA, okay? These are called the introns and they need to be cut out. Now, basically how you cut up the mRNA and then restitch it back together is not always set in stone. Sometimes there are different ways that you can do it, basically, okay? So for instance, sometimes there are exons which are optional, basically, i.e. you can include them or you might not want to include them, and you will still get a functional protein even if you don't include that exon. Yes, it might be missing a bit, but it will still be functional, okay? So, through means like this, you can actually get different proteins from the same gene, because you can end up with different mature mRNAs from the same piece of pre-mRNA here. Okay, so this is a mature mRNA. Okay, right, so basically this is the principle that you can have different splice variants that come from the same gene. And this process basically of turning a pre-mRNA, which is the primary transcript of the RNA, into a mature piece of mRNA by cutting out the introns and stitching together some of the exons back together. This is called splicing, okay? And it can lead to different mature mRNAs being produced, which will then lead to different proteins, and these different proteins are then referred to as the different splice variants of that gene. Okay, so certain of the genes for alpha subunits have multiple splice variants, which is how, even though you only have 16 genes, we can get 21 different subunits. Okay, right, so we will look at the genes rather than the subunits. So let's look at these 16 different genes for alpha subunits. So they are categorized into 4 families. Okay, so the first family is the G-alpha S family of alpha subunits of heterotrimeric G proteins. Now, there are 2 genes that are within this family of G-alpha S alpha subunits of heterotrimeric G proteins. There is the G-alpha S gene itself, okay, and then there is also the G-alpha ALF gene. Okay, so both of them are grouped into this same family. And what's special about these two is that both of them, when they're in the active state and have GTP bound to them, they both go off to activate adenylyl cyclase enzymes. Okay, so they both have very similar function. In a way, G-alpha ALF, named because it's found in the olfactory epithelium, is just the replacement for G-alpha S in certain places, such as the olfactory epithelium. Okay, so they're categorized into families together, and I want to stress these are genes coding for alpha subunits of heterotrimeric G proteins. The next family of alpha subunits of heterotrimeric G proteins is the G-alpha I/0 family. Now this contains 8 different genes, okay, so it contains 3 genes which can all often be called the G-alpha I's, okay, so there is G-alpha I1, then there is the gene G-alpha I2, then there is the gene G-alpha I3, okay, so those are the 3 G-alpha I's, okay. Then you also have some alpha subunits which are very similar to the G-alpha I's. Now, obviously they've all been put into the same family, so they are all quite similar, every single one of them that we're going to go through, but some of them are more similar to these G-alpha I's than some of the other ones that we'll see. Okay, so then you have G alpha Z, G alpha O. Okay, then you have two transducin G alpha subunits. Okay, so you have G alpha T1, G alpha T2. Okay, and basically these are the alpha subunits for the heterotrimeric G protein transducin. So in the retinal photoreceptors, such as rod cells and cone cells, basically the rhodopsin receptor, which we've discussed before, basically this becomes activated by light and will... Oh, well actually, have we discussed the rhodopsin receptor? No, we've discussed the rhodopsin family of G protein coupled receptors, but basically the rhodopsin family is named after rhodopsin, which is the archetypal member basically, and rhodopsin is a G protein coupled receptor which is activated by light. Okay, so basically when light hits rhodopsin, it will become activated and it will then interact with a heterotrimeric G protein, and this heterotrimeric G protein is called transducin. Okay, now the alpha subunit of transducin in rod cells is the G alpha T1. Okay, so this is the alpha subunit of rod transducin. Okay, and the alpha subunit of transducin in cone cells is G alpha T2. So this is cone transducin alpha subunit. Okay, right, so where are we up to now? We've done 7 of these. There's one final member of this G alpha I/0 family of alpha subunits of heterotrimeric G proteins, which is G alpha gust, which is involved in pathways in the gustatory system. Okay, right, so all of these have similar functions, but some of them have more similar functions than others. So for instance, all of these ones here, G alpha I1, G alpha I2, G alpha I3, G alpha Z, and G alpha O, they have very, very similar functions, okay? And we'll be coming back to these later. Okay, right. Then the next family of alpha subunits of heterotrimeric G proteins is the G alpha Q/11 family of alpha subunits of heterotrimeric G proteins. This contains 4 members. It contains the G alpha Q gene, the G alpha 11 gene, the G alpha 14 gene, and then also a gene which is called G alpha 15/16. Okay, now this is not 2 separate genes, this is one gene basically. Now let me tell you the story behind how it's come to be called G alpha 15/16. So basically people who were initially doing work in mice found a G alpha subunit which they called G alpha 15. Okay, and then when people did research in humans, they found G alpha subunit which they called G alpha 16. Okay, later it transpired that actually G alpha 16 is really the human version of G alpha 15. Okay, so it would be quite nice if we actually called these two the same thing. Okay, so we then renamed the G alpha 16 gene G alpha 15/16, and hopefully also we renamed the G alpha 15 G alpha 15/16. Now it hasn't caught on as well as people would have hoped, so you will often hear people referring to G alpha 15 if they're working in mice, and also G alpha 16 if they're talking about humans basically. So sometimes you will hear G alpha 15/16 called G alpha 16, especially when you're working with humans. Okay, then the final family of alpha subunits of heterotrimeric G proteins is the G alpha 12/13 family of alpha subunits, and this contains 2 members. It contains G alpha 12 and G alpha 13. Okay, so this gives us the 16 different genes for alpha subunits. Now, where do you get all the different splice variants from? Well, for instance, G alpha S has a huge number of different splice variants. G alpha O has 2 quite famous splice variants. It has G alpha OA and G alpha OB, which are just different splice variants of that same gene. Okay, so some of these genes have multiple splice variants basically, but these are the 16 different genes for alpha subunits of heterotrimeric G proteins. So let's just count them up. We have 1, 2, 8 here, 4 here, that takes us up to 14. 2 more, that takes us up to 16. Okay, so there are the alpha subunits of heterotrimeric G proteins. Now, let's turn our attention to the beta subunits. So basically, if we talk about the beta subunits, there are 5 genes for beta subunits in the human genome, and there are 6 different subunits. So we have 5 separate genes, and then 6 different subunits. Okay, so by now you should be able to predict what's going to happen. Basically, one of these genes will have 2 separate splice variants. Okay, so the 5 genes for beta subunits are called the beta 1, beta 2, beta 3, beta 4, and beta 5. Okay, now beta 1 gene produces one protein which is called the beta 1 subunit. Beta 2 produces one protein which is called the beta 2 subunit. Beta 3 produces one protein which is the beta 3 subunit. Beta 4 produces one protein which is the beta 4 subunit. Beta 5 then is the one with 2 splice variants. It produces 2 different proteins, one of which is called beta 5, and then the other which is called beta 5L. Okay, so these are the 2 different splice variants of the beta 5 gene, beta 5 and beta 5L. So that overall then gives us 6 different beta subunits of heterotrimeric G proteins. Okay, let's now talk about the gamma subunits, and thankfully the gamma subunits are extremely simple. Okay, so the gamma subunits, there are 12 different genes for gamma subunits, okay? And then there are 12 different subunits, so you don't have to worry about different splice variants for the gamma subunits. Okay, so what are the names for these different genes? Well, basically they are named very logically with one little blip, okay? So it's gamma 1, gamma 2, gamma 3, gamma 4, all the way up to gamma 5. Then there is a blip, there is no gamma 6. It goes straight on up to gamma 7, gamma 8, gamma 9, gamma 10, gamma 11, gamma 12, and then it has to go beyond gamma 12 up to gamma 13, and that's because we missed out the gamma 6 here. So that now overall gives us these 12 different gamma subunits. Okay, and these are the 12 different genes, and obviously they code for a protein which will be named accordingly. Okay, so all of this was basically to make you appreciate how many different heterotrimeric G proteins you could potentially build. There are 21 different alpha subunits encoded by 16 different genes that we've seen. There are 6 different beta subunits encoded by the 5 different genes that we've seen. There are 12 different gamma subunits encoded by 12 different genes. So there are a huge number of plausible combinations. Okay, so we want to now discuss which heterotrimeric G proteins do opioid receptors interact with. Well, the answer is we don't know exactly. We certainly don't know which beta and gamma subunits they specifically activate with. But the alpha subunits, what we do know is that the alpha subunits are certainly within this family of G alpha I naught, and they are most likely these 3 here, G alpha I1 to G alpha I3. Okay, but potentially you could also have other members of this G alpha I naught family also being activated by the opioid receptors, such as the G alpha Z, G alpha O, and potentially other ones as well. Okay, but most definitely these 3. So people will often just abbreviate these all to G alpha I rather than acknowledging the 3 different types. Okay. So, Okay, so the heterotrimeric G proteins that these opioid receptors are going to activate will have as their alpha subunit one of the three G-alpha I subunits, or potentially some of the other ones in that family of G-alpha I/naught. It's still not exactly known, basically. There's still a little bit of fogginess in the area, but we definitely know they couple through heterotrimeric G proteins with the three G-alpha I subunits, okay, as the alpha subunit. As for what beta and gamma subunit they use, we don't know at the moment as to whether there is any selection on that. It doesn't seem as though there is. It seems as though you can have whichever beta gamma subunit you like, okay? So people will often say that opioid receptors work through GI, heterotrimeric G proteins. Now what this tells you is it tells you the alpha subunit of the heterotrimeric G protein. And this is common nomenclature. People, when they're naming heterotrimeric G proteins, do not tell you exactly which alpha, exactly which beta, and exactly which gamma subunit you're working with. Very rarely do people tell you that. Okay, instead they don't even mention which beta and which gamma subunit you're working with, but they do tell you something about the alpha subunit. Now, generally they don't tell you exactly which alpha subunit you're working with, but they give you a good clue, okay? And when people put an I down there, they generally mean that it's got an alpha subunit, which is either G-alpha I1, G-alpha I2, or G-alpha I3. Okay, and we'll see the effects of these G-alpha I subunits in the next video.